Hello everyone, today we will give a brief introduction to the 2D GC mass. 2D GC mass is short for two-dimensional gas chromatography mass spectrometry. Here is the outline. First we will talk about the history and then talk about the experiment setup. And then I will discuss the principles of the 2D GC mass and then give a typical spectrum of the 2D GC mass. About 100 years ago, Golly invented a column. This kind of column can be used to separate the compounds in the complex samples. This kind of column is a column that we used in gas chromatography today. And 50 years ago, McLaughlin tried to couple the GC with mass spectrometry to increase the sensitivity of the method. And about 20 years ago, this guy invented the two-dimensional GC. That means we use two columns to separate the compounds in the very complex samples. That much increase the sensitivity of the analysis. Here is the typical experiment setup. First, the sample is injected into the injector through injector to the first column, and the first column is usually apolar, and the separation is based on the boiling points of the different compounds. And then after separation by the first column, the sample goes through the modulator, and this kind of modulator is used to isolate and reconcentrate the sample with a certain retention time, and then introduce the sample into the second column. And when the sample is injected into the second column, the sample is more completely separated by this column, because this column is polar, and the separation is based on the different polarity interactions. So this kind of two-dimensional column is called orthogonal configuration. Here is the more detailed principles. The figure A, first the first column separates the compounds and then can form a sharp band. And to the figure B, we can see this kind of sharp band is introduced into the modulator. This kind of introduction is by heating pulse generated by the modulator. And when the figure C, the sample passes through the modulator, the sample accumulates and then cool down. And the figure D, the sample is re-injected into the second column through another heating pulse, and then this kind of sample can be separated by the second column as be shown in the figure E, and then this kind of different compounds can be detected by the mass spectrometry. Here is the typical spectrum. In a typical one-dimensional gas chromatography, we can only, if we see one peak, this one peak may present three different peaks. That means this one peak is the convolution of three different peaks of three different compounds. But if we introduce the second column to invent the two-dimensional chromatography, then we can separate the three peaks in the second dimensional chromatography. And then by the typical transformation, we can form the 2D spectrum as we can see here. And then visualization, we can make the figure more clearly. The different color represents the different intensity of the compounds. On the right hand side, we can see is a typical spectrum. We can see we have two axes. The first axis, the y-axis represents the first dimensional retention time, and the x-axis represents the second dimension retention time. Here is a short summary.